Real world. Dead as a dodo. Program 2, transmission copy dubbed.
This man is having his head examined. Not that it'll bother him. He's dead, and has been for 8,000 years. What's amazing is that there is anything inside his skull at all. This is the oldest human brain ever to be investigated, and it's offering us vital clues in an extraordinary scientific detective story that will perhaps one day answer the biggest question of all. Where did we come from?
Who, or even what, were our earliest ancestors? Did we all emerge from primeval sludge? Or is there the fantastic possibility that we came from somewhere else, wafted here on a particle of meteoric dust?
And in the course of trying to find out where we came from, could there be a possibility that forms of life we once thought dead and gone forever could walk the earth again? Have we really seen the last of the dodo?
This recent headline from an American newspaper, famous for its exaggeration, may be a little too imaginative. But surely the same can't be said about this one from one of the world's most important scientific journals. DNA, the chemical foundation of life itself, cloned from a 2,000-year-old Egyptian mummy.
Tonight, we're going to try to unravel the mystery of our past. To help us do it, this unprepossessing chap. Actually, he's a bit of a mystery himself. At this end, he's clearly a horse.
Whilst at this, he is obviously more like a zebra. He is, or rather was, because he's been extinct 100 years, a quagga, surely one of the unluckiest of all animals, hunted into oblivion in the plains of South Africa only months before a law made him a preserved species. But he's achieved distinction in extinction, because he plays a historic part in our search for the past.
How life on this planet evolved to produce man has always fascinated us. But not so long ago, all we had to go on was just bones. And there is a limit to what a dry bone can tell anyone. But surprise, surprise, over the last few years, scientists have realized that these dried up remains still contain tiny amounts of protein. What are proteins? Well, they're the material that once filled these channels in the bone when the creature was still alive. Now, proteins are a vital ingredient not only of bones, but of the fleshier parts of the body, the parts which have long since disappeared. And finding out that proteins can still linger was a big breakthrough.
Within the last 6 months, it's been demonstrated in California that it's possible to extract even the original instructions from which these proteins were made. The very foundation of life itself, DNA, and that is going to tell us a great deal about where we came from. This is human DNA. In this sticky, viscous chemical are all the instructions which make up an individual, from the color of the eyes to the size of the feet. Now, most of us assume that when we die, this dies with us. So the first step in tonight's story was taken when scientists questioned that assumption. up the next link in our detective story here in the zoo in San Diego in California, where the animals are playing a part in helping to determine how life is as it is and where it goes from here. Quite painlessly, under anesthetic, as part of other routine investigations, small nicks are taken from many of the animals' ears, and these skin and blood vessel samples are then stored in a very different sort of zoo, a modern Noah's Ark, in which as many of the world's creatures as possible will be represented. Geneticist Dr. Oliver Ryder.
The frozen zoo was established to make the dwindling genetic resources of animals like endangered species available for future scientists to study. In some cases, we may not be able to prevent the extinction of species, but the essential genetic foundations, the germ nucleus of these species, can be preserved. But it's equally clear to us that we have no comprehension of some of the future uses to which these genetic resources might usefully be put.
But in 1982, Ryder received a sample he felt even he couldn't handle. This animal was extinct. There are a few photographs of one taken a century ago in London Zoo, but exterminated in its native South Africa in the 1860s, the very last Quagga mare died alone in the zoo in Amsterdam in 1883. Everyone thought that was that. But in 1979, whilst renovating a complete family of the animals which had been stuffed originally 150 years ago, a Quagga expert in South Africa had realized that the original job of preservation had in this case not been done very thoroughly.
Reinhold Rau of the South African Museum sent me the Quagga material, and he wondered whether the tissue would be worthwhile, but it was very clear when I got it it would be very useful because one could see veins in it. One could see, if you looked under the microscope, that there was the dried red blood of the quagga in these veins. And so I was optimistic that it would be possible to gain information from the proteins still in these samples about the question of what was the quagga.
A final answer to that was to be found in a laboratory across the bay from San Francisco in Berkeley. This is where Ollie Ryder sent some of his precious quagga samples, inspired by the work being done here in the Department of Biochemistry. Here, Dr. Russell Higuchi believed he could extract from the fragments of the extinct half-horse, half-zebra the most fundamental genetic fingerprint of all, DNA, the complex instructions from which all the different proteins that made up the living quagga were initially created. But if he could extract it, would it be in fit shape to be cloned, to be copied in the lab over and over again for research and investigation? He identified a solvent to strip out any DNA, introduced a virus to link up with any DNA, and added bacteria with which the virus, now carrying any quagga DNA, would react. Culture plates like this one revealed minute quantities of DNA were present, perhaps only about 1/1,000,000th of the total picture when the quagga was alive, but enough to be cloned, and enough to reveal a strong similarity to today's zebras and horses. In this case, a dot in the lower column indicates a match with a zebra. The comparatively few letters show where the DNA of today's zebra does not correspond with that of its extinct close relative. Higuchi had succeeded in extracting tiny but very valuable fragments of the chemical foundation of a life form that ceased to exist 100 years ago.
The kind of information we want could still be found in these very small fragments. And within those fragments, the sequence preservation was very good and accurate to the point where we don't think there is very much change at all, even though it's been dead for 100 years.
For Higuchi and his colleagues, this was just reward after considerably less successful work on tissue samples from Dima, a baby mammoth, found almost perfectly preserved after some 10,000 years in the permanent ice of Siberia. Up to now, the mammoth had resisted all attempts at DNA extraction, but the success with the quagga makes it well worthwhile working to increase the sensitivity of Higuchi's now proven techniques, because it's now clear that minute fragments of DNA, not just from the mammoth, but from creatures like the dinosaur, even longer extinct, might just be identifiable.
Russell's work is very significant. The quagga is but a single animal, however noteworthy. The greater impact is that we now, using the technology that he has developed so well, have the opportunity to go back and learn about the genetic structure of long-gone animals, whereas heretofore we could only base our analyses on what their appearance basically was. Now we can actually, theoretically at least, if we can find some dried tissue, or just a bit here and there, we can go back and find out what its genes were like.
The thing to remember in all this is the widely held belief that all life came from one original source and evolved in a variety of ways. So as scientists work their way backwards in time through the mammoths, even the dinosaurs themselves, we're getting closer to the original source of all life, including us.
Man's family tree stretches back through time, with our line branching away from those of our closest living relatives, the chimps, the gorillas, the orangutans, and so on, over millions of years. But exactly when the breaks took place has always been a subject of great argument. An argument we can now settle. Meet the next clue in our detective story. This is Ramapithecus. Now, he goes back 14 million years, and once upon a time, we thought he was a man. More recently, we've not been so sure. We have always known about the 14 million years, though, from his teeth, which were discovered. And when this was all the evidence we had, we thought Rama was more closely related to us than he was to any of the apes. And because we were absolutely certain about the 14 million years, the evidence from this meant that our family tree had to look like this. And there he is, Ramapithecus, fair and square on our direct line, clearly the earliest of men.
But today there is new evidence to hand. Not only have we discovered more bones and teeth, but scientists have started to study the proteins and more recently the DNA of living animals, including both apes and humans. And all of this indicates we're much nearer the apes than we thought.
In fact, if that new evidence is correct, all this is going to have to be redrawn. Now it looks as though we humans broke away from the orangutans at 10 and from the gorillas and the chimps a mere 4 million years ago. Why, this chap and I were as good as brothers.
And all that puts Ramapithecus in a rather embarrassing position. How can he go on pretending that he's a man when all the evidence suggests that he's, well, just another ape? Ramapithecus faced an identity crisis and took it to San Francisco, where he found a doctor who could solve his problem.
Dr. Gerald Lowenstein regularly analyzes protein from his hospital patients. In his research work, he's used the same standard technique to examine protein extracted from the teeth of Ramapithecus. If he could successfully compare the proteins of today's apes and humans with those of Ramapithecus, extinct for 15 million years, he believed he could help settle whose ancestor Ramapithecus is.
Ramapithecine proteins were no more like human proteins than they were like orangutan proteins or chimpanzee proteins or gorilla proteins. Therefore, my work strongly suggested what the earlier molecular work had also suggested, that Ramapithecus was an ape and not a man.
But I think your work was fundamental in achieving accord on this point, perhaps for the first time ever.
Yes, I believe that it was critical, but at the same time, people were finding other parts of Ramapithecus besides the teeth, and those other parts also strongly suggested that it was not human, but was an ape. So the new molecular data and the new fossil data happened to go right along with each other.
So poor old Ramapithecus isn't really one of us after all. So, what if we could examine not just the protein of long extinct creatures, but the chemical instructions from which those proteins were made, the DNA itself? The work on the quagga, extinct for 100 years, has proved that DNA survives long after death. The question is, how long?
The Egyptian obsession with preserving their well-to-do citizens as mummies has long fascinated those investigating our past. But a team from Uppsala in Sweden has just stolen all the scientific headlines by succeeding in cloning DNA from a mummy. Now, such remains are at least 2,000 years old, so DNA can survive a long time, but it takes persistence to find it. The Swedes tried no fewer than 23 mummies before they found any hint of clonable DNA. But even that achievement will be eclipsed by the next clue in our detective story. Last year, a team from the University of Florida in Gainesville, who'd been working for 2 years on a site near the Kennedy Space Center at Cape Canaveral, made what is almost certainly the archaeological discovery of the decade. A skull of a human female, which initial tests suggested was of the order of 7,000 years old. The following day, the skull of a 27-year-old male was unearthed. But it was only when these two vintage discoveries had been safely transported to the lab that the real significance of the finds became clear. During the standard wash and brush up to which all specimens like these are subjected, it appeared that there was more here than just bone and teeth. A routine hospital test followed, and it revealed that inside the skull there was something which looked like a brain. The back of one of the skulls was removed, and a silence fell over the lab as the team realized that it was looking at a human brain that had to be at least 7,000 years old. The quality of the discovery was later confirmed in tests with scanning machines and X-ray units. Through sheer luck, the deep, peaty conditions of the Florida swamp had preserved the tissue of the brain over the years, though one had shrunk to a third of its life size and the other was about 60% of the size one would expect. What mattered was within the fragments of soft tissue there might just be human DNA, much, much older than anything ever worked on before. DNA which could be extracted. In the lab, the Gainesville team set to work on what are almost certainly the oldest preserved human brains in the world. The DNA strands from the two brains, now certified as around 8,000 years old, are readily identifiable. They are clearly human. They are remarkably intact. But the supply of material like this for research is obviously limited. For the important, detailed detective work that can now be done on our distant ancestors, this evidence has to be cloned, to be copied over and over again.
Dr. Phil Lapis is one of the Gainesville team, and he's flown over to join us on The Real World. Phil, can I ask you to describe that moment when you realise what you've got isn't just a lump of earth clogging up the skull, but a real brain?
There was just dead silence in the lab. I was holding what was recognizably a human brain. I mean, everyone had seen one, and we knew what we had was an 8,000-year-old brain. It was total amazement. We had not expected that at all.
What have you actually done with the brain since that moment? What have you discovered about it?
There's a surprising amount of structure left in the brain. There are some cells still intact. We've been able to isolate DNA from that brain. We've shown that at least some of the DNA is human DNA. And we've been able to clone some small fragments of DNA from the brain.
What is the next step?
What we have to do is look at the DNA fragments we have. We have to get more of them and match them to known pieces of human DNA. It's as if we had a sentence out of Hamlet. We didn't know where it belonged, and we were trying to match it to the whole volume of Shakespeare. We have those individual lines. We're trying to match them now, to look for the to be and not to be soliloquy in Hamlet.
But what is its real relevance? What are we going to learn from all of this?
We're going to learn, or the archaeologists in particular are going to learn, where these people came from. They may be able to learn whether these people left any descendants. From my point of view, we're going to learn something about how DNA survives in the environment after the individual is dead, how much damage occurs, how much it has changed, both from damage and just from the normal evolutionary processes that has happened over 8,000 years.
Do you think that one day you may actually be able to find out things about samples of DNA that are even older
I think so. We know how much damage has occurred here. I think that we'll be able to go back another perhaps 30,000 years, perhaps a little bit longer, and if the DNA is as well preserved, we'll be able to clone that DNA as well, and that may be much more interesting to me.
Sounds it. Thank you very much indeed.
Should really elderly DNA prove viable, then that will bring to a positive conclusion perhaps the most intriguing element of all of this story. It concerns a fruit fly, happily buzzing through the sunshine, who decided to take a rest on the bark of a passing tree. It wasn't a very good idea, because that particular part of the tree was covered with gluey, oozy resin, and the fly stuck fast. All this happened 23 million years ago in what is today the Dominican Republic. But over the years, the resin hardened into amber, encasing the fly in a perfectly preserved time capsule. Today, entomologist George Poyner, working at Berkeley, believes that the evidence contained in this earliest of known preserved life forms could, thanks to the techniques now perfected, prove extremely exciting.
The specimens that we have found in amber are interesting for two reasons. First of all, they tell us what existed on Earth 20 to 40 million years ago, so we can have an idea on what kind of insects occurred here and how they compared with present-day forms to give us some idea of the evolution. Then they're interesting from the cellular standpoint because possibly we can revive some of the cells because we do find them in remarkable condition. In other words, we could possibly find DNA still inside the nuclei of some of these flies preserved in Baltic amber, which is 40 to 60 million years old.
Are you confident that the DNA in that amber is viable?
Well, if we found that it was viable, this would open up a new chapter because no one really believes that DNA can be viable for such a long period. So we wouldn't confirm anything that we already knew. We'd be opening a new area and providing some new evidence as to the longevity of nucleic acids and possibly even in a broader scope the origin of certain forms of life here on Earth. Because if the DNA could remain viable for up to 40 million years, there's an indication that this is a time period in which material could come from other planets, from other extraterrestrial sources, and be brought here to planet Earth.
What George Poyner is saying is that if the fruit flies prove that DNA can survive 60 million years, that would be long enough for some primitive life form to make an exceedingly long journey, arriving on Earth on board one of these, a meteorite from space. If all life came from one original source, that is a real eye-opener.
Mind you, it's not a new suggestion. One eminent British scientist, Professor Fred Hoyle, has been claiming for years that life came from space. Who knows? Perhaps Sir Fred is right. And a fruit fly could confirm a link with the past, which we all share somewhere beyond the stars.
And from one idea, which up to now has been regarded by most as a complete flight of fantasy, how about the equally incredible possibility that if we can extract DNA from quaggas, mammoths, even dinosaurs, perhaps one day we might reconnect those complex chemical chains so that creatures we once thought extinct could once again walk the earth. Can we any longer say with certainty that that will never happen?
I wouldn't say never. We might get lucky. We might find the sample that's been perfectly preserved, and we could have woolly mammoths going through Rocky Mountain National Park again. I'd like to try it.
I can't foresee how this could come about in the future, but scientists are notoriously poor predictors of what the future will hold.
I think it's entirely possible, but obviously we can't do it unless we have a program for a quagga or a mammoth, which consists of the total amount of DNA for those creatures. And so I think it behooves us to store that information, and being able to clone them is just the technical problem. It may take another 20 or 30, even 100 years, but I think eventually we will be able to do it.
Now, if any of you find all this a little complicated, well, it is. But just remember, what it boils down to is this. The next time you find yourself face to face with a dodo, remember, we did warn you, here on The Real World. Good night.
scientific program to revive the extinct quagga is making good progress. The last quagga, related to the zebra and once common in South Africa, died out in 1883. Apart from a breeding herd established in the Cape, the fund supporting the work was given a boost with a donation from the organized hunting community in South Africa. The donation was made in the interests of game conservation.
Well, what we're trying to do is to retrieve the genes from the quagga from the extant population of Birchall zebra. The way we're able to do this is based on the fact that molecular studies from material which was originally sent across to America some years ago to scientists in San Francisco have shown that the quagga is in fact very closely related to Birchall zebra. So closely related that you can in fact say it's no more than a subspecies of most. There's very little difference. Therefore, there's no reason why we shouldn't be able to find the genes which characterize the quagga, namely the genes which account for the faint degree of striping over the rear end of the quagga and the darker color which characterize the quagga, to find those genes amongst the Birchall zebra and therefore recreate or retrieve these genes into the an inbred population by selective breeding. In this way, we will hopefully be able to show the public eventually a quagga. And in fact, it really will be a quagga. It won't be an imitation quagga. It'll be, in fact, the real thing. Because if, in fact, the genes are simply dispersed in the current Birchalls or plain zebra, then we should be able to get them back again. And therefore, have as good a quagga as there ever was originally.
Great project. Let's wish them luck.
Diere. Die een hier aan my linkerkant is die bloubok wat net in die Kaap-provinsie voorgekom het, maar wat sommer baie gou na die koms van die blanke met sy geweer van die aardbol af verdwyn het. Die een daar agter is die baie bekende Kaapse quagga wat hier so aan die einde van die vorige eeu ook deur jagters en boere tot uitwissing gedryf is. En op beide van hierdie skilderye of afdrukke staan daar drawn and engraved by Samuel Daniels. Maar daar is nou 'n paar wetenskaplikes wat sê dit is wel moontlik om die oorlosie terug te draai. Hulle sê ons kan weer eendag egte Kaapse quaggas op hierdie aardbol hê as hulle planne net werk.
Die Kaapse quagga is dood. Op die 12 Augustus 1883 het die laaste een in die Amsterdamse dieretuin gevry. 100 jaar later het die Hollanders die tragedie met 'n spesiale posseël herdenk. Al wat vandag oor is van die duisende quaggas wat voorheen 'n algemene verskynsel in die Karoo was, is 'n paar opgestopte velle in Europese museums. Die Mary, wat 21 jaar lank in die Londense dieretuin was, is die enigste quagga wat ooit lewendig gefotografeer is. In die Suid-Afrikaanse museum in Kaapstad is daar daarom een opgestopte vulletjie, sodat ons ook kan sien hoe die quagga gelyk het. Die oorheersende kleur was 'n warm bruin met opvallende strepe oor die kop en nek. Verder agtertoe verdwyn die strepe sodat die lyf 'n effe vaalbruin is, amper soos die van 'n donkie. Reinhold Rau is die taxidermist by die museum wat die vulletjie gerestaureer het. Vir hom was dit nie genoeg om net die vel weer soos 'n lewendige dier te laat lyk nie. Hy het ook daarvan begin droom om die Kaapse quagga uit die bont sebra terug te teel. Jarelank het hy mense probeer oortuig dat dit moontlik is, maar niemand wou hom glo. In 1984 is Rau reg bewys, toe dit bevestig is dat die quagga en die bont sebra dieselfde diersoort is.
Scientists have been speculating about these possibilities for quite a long time, especially since the Heck brothers in Germany rebred a form of cattle which very closely resembles the wild ancestor of the European domestic cattle, the auerochs, which is extinct. Then in 1969, this museum decided to remount our little quagga foal, which was in a very poor state, and in doing so we found that the skin had not been cleaned from the flesh side, from the inside, very carefully. Portions of connective tissue and even muscle fibers and blood vessels with dry blood in it were still attached to the skin, which were then removed carefully and stored, and in the hope that somebody might be able to do some microbiological tests with it.
Uit die horingdroë stukkies weefsel is genoeg DNS gehaal om die quagga se genetiese kode te ontsluit.
DNA slowly accumulates changes, mutations, with time, in a rate which is steady and increases proportional to time. Now, they were able to sequence a little bit of the DNA which they'd extracted from this quagga tissue and show that the sequence of the DNA was almost identical to that of the plain zebra.
Die werk is in Amerika in die laboratoriums van Ryder en Ikuchi gedoen. Hulle het met elektroforese die volgorde van die basisse in die quagga se DNS-molekule uitgewerk. Die DNS word op 'n jelplaat gedruk, waarin die basisse van mekaar skei. Die plaat dra die dier se erflike geheime in kodevorm. Die DNS-molekuul bevat 'n dier se familiegeskiedenis. Daar is 'n lang spiraal waarop die 4 basisse in 'n kenmerkende volgorde gerangskik is. Die 4 is vir alle organismes dieselfde, maar die volgorde waar hulle gerangskik is, sal bepaal of dit 'n olifant of 'n muissinne is. As ons nou weer na die DNS-molekuul kyk en ons verwerk die inligting wat met die elektroforese metode gekry is, kan ons die volgorde van die basisse in die molekuul lees. Om diere se verwantskap met mekaar te bepaal, skryf ons hulle basisvolgordes neer in vergelyking. 'n Kol beteken ooreenstemming en 'n letter 'n verskil. Hier is soort B nou verwant aan A, maar C is net ver langs familie, want daar is min ooreenkoms en baie verskil. 'n Vergelyking tussen die Kaapse quagga en die vlakte zebra is net 'n rykolle, wat vir ons sê dat hulle genetiese vingerafdrukke presies eenders is. Dit beteken dat hulle meer as naby familie is. Hulle is een en dieselfde diersoort. Op een plek klop dit nie. En dis waarskynlik net omdat die weefsel so oud is.
It showed, in fact, that the quagga was so closely related to the plain zebra that you could say that to all intents and purposes it was a plain zebra. In other words, it was as closely related to the plain zebra as, say, a Siamese cat is to an ordinary tabby. Now, the implications of this is as follows. It means that you can validate a rebreeding program for the quagga because you can say that, in effect, the quagga is not extinct at all. It's all that you've lost is the color genes which characterize the quagga.
Equus burchelli, die vlakte zebra, is dus 'n quagga. En die naam bontquagga, soos hy van vroeg af bekend was, pas hom goed. Daar is 'n groot variasie in die kleur en streeppatrone van die bontquagga. Gewoonlik kom die strepe onderom die pens bymekaar en op die bene gaan dit af tot by die hoewe. Die bruin skaduwstreep word duideliker van noord na suid deur die bontquagga se verspreidingsgebied. En dan kry jy soms die enkeles wat min strepe het, sodat veral die agterlyf amper effe kleurig is. Die bene het dikwels glad nie strepe nie. En op die lyf kom die strepe ook nie onder die pens bymekaar, soos by die meeste ander bontquaggas nie. Dit is hierdie diere wat nog baie van die Kaapse quagga se gene dra en wat nou vir die teelprogram uitgesoek word. Die Natalse Parkeraad werk ook saam in die program en hulle wildvangers is altyd op die uitkyk vir die regte diere. As hulle met roetine wildvangste enige bontquaggas met die min strepe oor die agterlyf en bene sien, word hulle net daar verdoof en gevang. Diere wat met dwelmpiltjies geskiet is, gaan lê soms op ongemaklike plekke, ver van die vragmotor wat hulle moet oplaai. Hierdie draagbaar met 'n wiel is 'n Natalse uitvinding wat wildvangery baie makliker en veiliger maak. Die dier kan daarmee baie vinniger teruggebring word, wat die gevaarlike tyd terwyl hy onder verdoewing is korter maak. Die diere wat in Natal gevang is, word vereers na die Gloeloe wildtuin gebring, waar hulle in wildhokke gehou word. Hier kan hulle nou rustig en van naby bekyk word, om te sien of hulle regtig goeie teelmateriaal is. Wanneer daar genoeg diere is, kom Reinhold Rau die diere uitsoek wat hy wil gebruik. Die Vrolikheid Natuurbewaringsstasie is naby Robertson in die Suid-Kaap en lê in die voormalige verspreidingsgebied van die Quagga. Dit is hier waar die teelery gedoen word en waar die eerste Kaapse Quagga in hierdie eeu hopelik gebore gaan word. Geskikte teelpare word in kampies aangehou. Daar sal daarom nie twyfel wees oor die vaderskap van die filletjies wat gebore word nie, en akkurate rekords en stamboeke sal opgestel kan word. Een van die eerste take is om die diere aan mense gewoon te maak, sodat daar later makliker met hulle gewerk sal kan word. Bondquaggas teel stadig. Merries kry hulle eerste vullings eers wanneer hulle 4 of 5 jaar oud is. En dan is die dragtigheidsperiode net meer as 'n jaar lank. Wanneer die diere makker en hanteerbaarder is, word beplan om met kunsmatige inseminasie en met embryo-oorplantings die proses te versnel. Die herteling van die Quagga is 'n gesamentlike projek van die Suid-Afrikaanse Museum en die Kaaplandse afdeling Natuurbewaring met die samewerking van die Universiteite van Kaapstad en Stellenbosch. Daar sal klaar 'n hele paar teelpare by Vrolikheid, wat die baie beperkte streeppatroon op die agterlyf het. Daar is nog 'n tekort aan die ander eienskap waarvoor geteel moet word, die bruin agtergrondkleur. Ook hier is hoop op pad.
Ons sal baie bepaald graag betrokke wil wees. Ons het ook ons hulp aangebied. En waar ons in ons groot troppe quaggas in die Kruger Wildtuin bepaalde afwykings sien wat veral die bruin op die rug, wat baie belangrik is vir die terugteel van die uitgestorwe Kaapse Quagga, wat daai bruin vlek op die rug toon, sal ons hulle uitvang en vir ons kollegas in die Kaap aanbied.
Voordat de mens te vinnig op hol gaan oor die hele projek, en wat daaruit gebore kan word, is het goed om dalk eers 'n bietjie stil te staan en na te dink. Want daar is tog ook wetenskaplikes wat nog nie heeltemal oortuig is nie.
Wel, dit is 'n heel interessante oefening. Dit bevestig wat baie mense lankal gevoel het, dat die uitgestorwe quagga in werklikheid net die suidelike bevolking van die bondquagga is wat verder in Afrika voorkom. Dit sal egter nie 'n quagga wees wat herskep word nie. Die quagga is uitgesterf. Die seleksie wat plaasvind is net vir die velkleur en velpatroon, terwyl 'n quagga in werklikheid uit baie meer as net dit bestaan het. Hy was aangepas by sy spesifieke omgewing daar in die Karoo. En dit sal dus 'n mensgemaakte dier wees wat gaan lyk soos 'n quagga. Ek wil graag aanhaal wat meneer Hokko Knobel, die destydse hoofdirekteur van Nasionale Parke, dikwels gesê het. 'n Mens kan nie skep nie, hy kan nie herskep nie, hy kan net bewaar.
If in fact Ryder Noguchi's work had shown that the DNA was very different between the quagga and the plain zebra, this sort of rebreeding program would be quite unjustified. But the fact, in fact, that they've shown that the quagga and the plain zebra are the same species means to say that we will not just be rebreeding a quagga look-alike, we will really be rebreeding a genuine quagga, since the only thing that defines a quagga are these color patterns, and since the genes are probably simply diluted out in the extant, that's the current population of plain zebra, it should therefore be not only possible to get these genes back by a careful selective program, but the animal you produce at the end of the day will be a real and genuine quagga.
Die Nasionale Parkeraad as nasionale bewaringsliggaam is natuurlijk besonder opgewonde oor die moontlikheid dat daar 'n uitgestorwe species sal teruggeteel kan word uit genetische materiaal wat steeds beskikbaar is in populasies van die vlaktezebra. En ons sien met groot opgewondenheid uit na die dag wat hierdie diere weer op die Karoo vlaktes en die res van hulle verspreidingsgebied sal kan gesien word deur die publiek van Suid-Afrika.
effects on man and the environment. From Brazil, where man threatens man in the rush for modern wealth, to Britain, where a priceless collection falls victim to capricious nature, and to Australia, where a huge river is slowly dying. Some of the reports on Earthfile. First to South Africa, where scientists rebuild a long-dead animal.
When the early settlers pushed inland from Cape Town, capital of the Cape Colony, they encountered, to their delight, vast numbers of zebra. Great herds of one particular zebra subspecies, the quagga, roamed freely across the Great Karoo Plains of South Africa, from Bushmanland in the west to the Orange Free State in the east. But the quagga, now regarded as a close relative of these southern plains zebra, was doomed. Ruthlessly slaughtered by the new settlers for meat for their Hottentot workers, the ill-fated quagga never had a chance. By the year 1883, it had become extinct, a museum piece, yet another reminder of man's lack of foresight. This mare died at the London Zoo in 1872. Ironically, the hunting of the ill-fated quagga and other threatened animals was finally outlawed, just 3 years after the very last quagga died at a zoo in Amsterdam. But this man, Reinhold Rau, the chief taxidermist at the South African Museum, is convinced that the quagga can be virtually reincarnated, not by divine intervention, but through a breeding program involving stock carefully selected from the quagga's cousins, the southern plains
since this is
zebra.
now confirmed, there is of course good reason to believe that quagga genes are still alive in the present southernmost populations of the plains zebra.
While reconstructing the remains of this quagga foal, the taxidermist noted the similarity of its color and stripings to the markings of related living zebra.
The stripe and the interspace is usually roughly the same width. In the quagga, the interspace, or the light stripe if you want to, is usually narrower than the actual stripe. Now we select for these three characteristics: narrow interspaces, dark basic color, and reduced striping.
As a result of Mr. Rau's findings, an ambitious rebreeding program has been launched here by the Cape Department of Nature and Environmental Conservation. Dr. Jeff McLaughlin has carefully supervised the capture of 8 plain zebra from the Etosha Game Reserve in Namibia. But the program has been extremely controversial.
Several people had suggested trying to breed plain zebra without as much striping as they normally have. But a lot of people objected to this because they said, well, they didn't know what the quagga was originally. They thought it was perhaps a totally separate species, and therefore there was no point in making a thing. All you would get is a look-alike that you could say looked like a quagga, but it had no real relationship to the extinct animal.
But the question of relationship was resolved by a Californian biochemist, Dr. Russell Higuchi. He believed he could extract from a fragment of the quagga the most fundamental genetic fingerprint of all, DNA. Deoxyribonucleic acid. The complex instructions from which all the different proteins which made up the living quagga were initially created. But even if he could extract the DNA, could it be cloned for further essential research? Dr. Higuchi identified a solvent to strip out any DNA, then introduced a virus to link it up and added bacteria, with which the virus now carrying any quagga DNA would react. Culture plates like this one revealed minute quantities of DNA were present, perhaps only about 1/1,000,000th of the total picture when the quagga was alive, but enough to be cloned and enough to reveal a strong similarity with today's zebras. In this case, a dot in the lower column indicates a match with a zebra. The comparatively few letters show where the DNA of today's zebra does not correspond with that of its extinct close relative. Dr. Higuchi had succeeded in extracting tiny but extremely valuable fragments of the chemical foundation of a life form that ceased to exist a century ago.
The kind of information we want could still be found in these very small fragments. And within those fragments, the sequence preservation was very good and accurate to the point where we don't think there is very much change at all, even though it's been dead for 100 years.
The scientists' findings ended the arguments about the relationship of the quagga to other zebra. Clearly, it was the southernmost representative of the plain zebra, a subspecies which had lost its striping simply because it didn't need it.
So now the whole experiment made sense. It was worth doing because if we produced something, it would be genetically related to the plain zebra and therefore a genuine quagga, not just some sort of look-alike which you might have got by crossing a donkey with a zebra, something like that. But that would have been a meaningless exercise.
With luck, the breeders say they could produce a quagga within 3 years. But more than likely, the program will take at least a decade. However long it takes, Dr. McLaughlin and his team are convinced the work must continue. They're determined to reintroduce an animal needlessly and shamefully exterminated by man.
As most of us know, this animal, the quagga, became extinct at the beginning of the century, and that was as a direct result of man's actions. This, together with the extinction of the bluebuck and the Cape lion, will remain an indictment against us for allowing it to happen. But a few years ago, a scientist made an astonishing claim. He said, all is not lost. A project was started a few years ago to rebreed the quagga. Well, we screened a report on this at the time, but what has happened subsequently? Belinda has the story.
About 200 kilometers outside of Cape Town is the Frolicape Nature Reserve just outside of Robertson, and it's here that a breeding program is currently underway. The objective of this breeding program is to try and bring back the extinct quagga. The quagga got its name from the Hottentot because of its kind of quagga bark. It's striped on the forequarters like our ordinary zebras, but the hindquarters look more like a chestnut donkey. Now, it's difficult to prove, but there are suggestions that the quagga was not a one-off species, but in fact a member of the horse family, which underwent a process of evolution.
This, let's call it an academic venture, started when Mr. Reinhold Rau, a taxidermist at the South African Museum in Cape Town, was mounting an old skin of a quagga foal. Musing over an experiment conducted 50 years ago, he set up a scientific committee to study the evolution path of the quagga and to look into the possibility of doing a similar experiment. But just how do you study an animal which has been extinct for almost a century? The only living close relatives are, of course, horses, donkeys, and zebras. But just how different is the quagga from, say, a zebra? Dried blood and muscle tissue were removed from the quagga skin and sent to the University of California, where it underwent DNA analysis by 3 American scientists. The DNA molecule contains an animal's family history, as it were, in code form, in which 4 bases are arranged in a particular order. The arrangement of these 4 basic elements will determine what the end result will be, either a mouse or a quagga. A comparison between the DNA of Birchley's zebra and the quagga revealed that it matched almost perfectly, but for one symbol. But the experts say that the age of the sample could have influenced the result. On the strength of this test, they argued that the gene pool for producing a real-life quagga must still be here in South Africa. For them, it was simply a matter of matching animals with the right potential in a breeding program. The project really got going when the first 9 animals arrived at Frolicape in April 1987.
The Frolicape Nature Reserve was chosen as a breeding site for a number of reasons. Apart from the ideal climate, it's also in an area known as the Bar Valley, well known for the breeding of racehorses. And it's this that would have provided the ideal veterinary and medical backup. In early 87, a number of large paddocks were built for the animals, but it was in March 87 that the breeding committee actually went out to capture selective zebras.
Over the next few years, animals from as far afield as Zululand and Etosha were caught for the quagga project. They carefully selected only animals that potentially carried the correct genes. That meant animals had to have very light markings on their hindquarters, but this was not the only criteria. The dark brown shading was also a factor considered. The project was at first met with some skepticism in certain circles. It also stimulated a lot of interest among scientists and conservationists alike. Once they realized it was not just an idea, information about the whereabouts of potential breeding stock started coming in. Then it was just a matter of roping them in. Of course, a lot of respect was given to the safety and care of the selected animals. The capture teams carried out the operations in the most humane way possible to minimize the stress of the animals, a factor responsible for most deaths during a game catch. Remember, history was in the making. Once the zebras were delivered safely to Frulicate, they were left alone to get used to their own environment. All they had to do now was to breed, eat, and breed again.
From the project's humble beginnings in 1987, they had the birth of the first foal in December 1988. Joxy, unfortunately, didn't show much of reduction in her striping. The second foal died because of natural causes. The third foal, Emma, was born also without much reduction in her stripes, but then she was also used to breed for colour. And the fourth foal is where scientists really got excited, because now she, Rhino, had a marked reduction in her striping.
The Quagga project also generated a lot of interest amongst the international scientific community. Even the renowned Professor Rufus Churcher, a Canadian scientist, ventured out to Robertson to witness this historic event.
I think this one is quite unique. It is a unique program because we are trying to bring back an animal that is effectively gone, except the genes are still around. The Poles did something of the same sort before the Second World War in bringing back the aurochs, the wild ox of Europe, from the more primitive breeds of domestic cattle. It's the same sort of thing. We're trying to select genes and thus conformation of an animal that isn't around at the moment, but we believe it's still in the populations that exist.
With the project established and having had limited success, hopes have now been raised that the Quagga could in fact be rebred. But let's say this does happen, what then? Dr. Kluwer, head of the Quagga breeding project, feels that it is more than just a pipe dream.
Once we've reached that stage, I think we will have crossed a threshold, as it were, in the whole undertaking. And what we would like to see there is at least a reserve set aside for these animals to live in and to continue to breed. In that sense, they will be a new resource for humanity in the highest sense of the word. Really, we would have put something back into South African and in fact into world fauna, something which has disappeared and which is now in a way being brought back again, not recreated but just brought back.
An aspect that cannot be ignored, of course, is whether the Quagga is truly being rebred or whether a look-alike is merely being achieved. From a scientific point of view, it's an aspect that in all fairness must be considered. Professor Heidenreich, also a project member, is opposed to the term look-alike.
People interested in the Quagga in other parts of the world are not on the forefront with the newer developments since 1984, and many of them are opposed to the project. And I think that the work published by the Americans as well as by the South African Museum and others will in the end prove to people that there can be no doubt that the Quagga was in fact a subspecies of plain zebra and that we are on the right track.
Contrary to what was expected, some of the offspring have more stripes than their parents and some have less. But scientists aren't too worried about this. Professor Eric Harley of UCT is a DNA and genetics expert and also member of the breeding committee. He's quite happy with the results.
The interesting thing about the foals that we've had is that some of them are more striped than the parents. In other words, they're going in the wrong direction from our point of view, but some of them are less striped. Now, that has already told us something new. It tells us that indeed there's a lot of variation in the genes, and the biselective breeding, we can either concentrate the effects in whichever direction we like, so we have to allow chance to produce either more striped or lesser striped species. So having already achieved some animal progeny from our breeding stock which are lesser striped than before, it suggests that the project should be successful, and that by continuing this process we should indeed end up with an animal sufficiently unstriped to be able to have all the characteristics of the original Quagga, at least in terms of the striping pattern.
The extinct Quagga evolved over thousands of years in the Karoo with its harsh environment. The fact is that various things caused the Quagga to change its diet and its look so that it could survive out there. Because it did adapt to its environment, it did manage to survive, until man came along. The Birchall zebra prefers grasslands. Would a grassland animal bred only over a period of decades to look like a Quagga learn so quickly to survive in the Karoo? Rufus Churcher thinks it is possible. After all, he says, the Przewalski horse and the European oryx were also once extinct. They have now been rebred and are doing well.
What this program will do, if it's effective, and I expect it to be so, is to produce animals that are ecologically similar to Quaggas, at least in color, And then if you introduce them into areas where you've got zebras and where the quokkas were, presumably they'll fill the niche and they'll look more appropriate, at least, than having a mountain zebra or a common zebra there.
At the end of 1990, another potential breeding animal was identified in the Sasolburg Nature Reserve. The mare only had a few stripes in the hindquarters, and Natref decided she must be donated to the Frulicate project to improve the gene pool. Dr. Jaime Ebedes of the Department of Agriculture, the former National Zoological Gardens veterinarian with many years' experience in game capture, was specially asked to coordinate the relocation project. The darting was done only after the zebra became accustomed to the presence of the vehicles, so that it wasn't necessary to dart her while on the run and in a stressed condition. She fortunately responded well, not at all like her companions. Stress remains the #1 killer of captured animals, and all sorts of plans have to be made to counter this. The mare had to be blindfolded and properly sedated for the 14-hour journey down to Robertson. Once she was safely loaded in the horse trailer, she was given an antidote to wake her up, as she couldn't be transported lying down all the way. The tranquilizer that she was given would keep her calm for the long journey ahead and prevent her from injuring herself.
We used a drug called etorphine, commonly known as M99, which is a morphine derivative, and it's highly effective in zebra, plus a tranquilizer. It's the most important immobilizing drug used for antelope and for zebra.
But sadly, the journey wasn't even half completed when the team discovered near Colesberg that she had died, despite all the careful precautions that were taken. The sad news only reached the breeding committee the following morning. This stroke of bad luck served as a grim reminder that there was still a long road ahead with a growing number of pitfalls. If they were to succeed, it would have to be done in the face of considerable odds. Apart from the physical problems such as disease and injuries, there are also factors such as the right choice of animals to complete this genetic engineering feat, which staunch conservationists and scientists such as Dr. Neil Farrell consider to be a waste of time.
Well, they can breed a lookalike, and the lookalike is going to have some value in showing people, look, there was something like this, isn't there anymore. But in terms of breeding a real quagga that we can put in a nature reserve and say that is a quagga, impossible.
Yeah, okay.
Dr. Farrell's objections are based on the fact that the committee is using data obtained only from the female strain of DNA and not the combined result of male and female. In other words, they can only be 50% correct. And it's this discrepancy that Dr. Farrell questions.
Because of the differences and because we don't really know what the quagga was. We didn't have time to study it before it died out.
Dr. Farrell says that nature's quagga, which was created through a very long evolutionary process, would also be genetically different from this New World lookalike. But Professor Harley differs.
The point we are making is that we are not trying to recreate an extinct species. Our point is that the quagga is not extinct, or at least we hope it is not extinct. We believe that the only things which characterize the quagga, shown to be the same species as the plain zebra, is the characteristic color patterns, which are simply the consequence of a few individual genes. Just as a Siamese cat is not a different species of cat, it's just that a rather small number of mutations, in fact really only one in the case of the Siamese cat, has produced the color patterns characterizing the Siamese cat.
Another problem might be inbreeding. By isolating a few animals in a small area, you are in fact narrowing their gene pool. Research done at the Rand Afrikaans University under Dr. Herman van der Bank has proved that inbreeding amongst family members also has its disadvantages in wild animals. The loss of genetic diversity leads to weaker animals that are more susceptible to disease. Is the death of one of the 5 foals so far an indication of this?
In 95% van die gevalle kry mens dat die diere by jong ouderdom sterf en in 75% van die gevalle kry mens dat die nakominge baie minder is. Met ander woorde, daar is alreeds baie groot mate van steriliteit. Maar dit wil tog voorkom asof hierdie mense probeer waak daarteen deur ook aparte eksperimente loop en uiteindelik gaan hulle die 2 verskillende so wat maar rasse noem weer kruisdeel.
The fifth foal, born in April this year, was still showing considerably more stripes than the only example of a young quagga that we have at the South African Museum. A sure indication that a lot of wife swapping must still be on the cards for the zebra family at Fruilicate. The question to ask then, is all this really necessary?
Necessary is a difficult question. Is nature conservation necessary? We say, of course, it is necessary for man's own survival, because if the environment is interrupted and disturbed, then it has bad effects on the continued existence of man himself. I don't know whether it is tragic in that respect, whether the quagga is gone or is back. But just for ethical reasons, since it was a mistake to bring it to extinction, we see it as an ethical duty of present-day men to try and rectify this mistake now, since the possibilities are given.
Who knows, with a bit of luck, we just may bring back the quagga to the Karoo. The fact is that we should never have allowed this animal to become extinct in the first place. The attempt at rebreeding the quagga is another lesson to man never to willfully allow the extinction of another species. They are truly just too valuable to lose.
Een prachtig oud klein ding diep.
Löwen kennen die Taktik ihrer Beutetiere. Sie müssen sich etwas einfallen lassen. Hier lauert ein Meister der Pirsch. Weder Antilopen noch Zebras bemerken ihn zunächst. All seine Mühe war umsonst. 5 von 6 Mal setzen Löwen vergeblich zur Jagd an. Ein erfolgreicher Versuch, hier in Zeitlupe aufgenommen. Ein faszinierendes Schauspiel. Dass Zebras aber auch dann durchaus noch eine Chance haben, zeigen diese Bilder. Erstaunlich, dass sie von solchen Verletzungen genesen können. Dieses Tier wird nie wieder so aussehen wie früher. Seine Fellzeichnung hat sich verändert. Eine große Wunde ist abgeheilt, die Streifen aber passen nicht mehr richtig zusammen. Solche Blessuren sieht man an vielen Tieren. Auf der Suche nach der fettesten Weide wandern die Zebras in der Trockenzeit in großen Herden durch die Savanne. Sie fressen, was andere stehen lassen. Das erste Raugras ebenso wie alte Strünke. Das ist wie Rasenmähen. Zarte Triebe können rascher nachwachsen und damit ist die Weide für die Antilopen vorbereitet, die nur weicheres Gras fressen. Obwohl die weite Etosha schon recht trocken ist, wird Wasser für die Wildtiere eigentlich nie zu einem Problem. Millionen Liter lagern tief unten im Kalksteinbett der riesigen Senke. Sie speisen Brunnen und einige Quellen sprudeln auch in der trockensten Zeit. Aus ihrer Nahrung können Zebras nicht sehr viel Flüssigkeit gewinnen. Deshalb müssen sie wenigstens einmal am Tag trinken. Zum Wasserloch und beim Weiden führt die erfahrenste Stute ihre Familiengruppe. Der Hengst gilt trotzdem als Oberhaupt. Innerhalb der Gruppe sind die Bindungen sehr eng. Am erstaunlichsten die zwischen Vater und Söhnen. Sie ist so stark, dass er die jungen Hengste auch dann nicht vertreibt, wenn sie schon alt genug für eine eigene Familiengründung sind. Allerdings kommt das nur selten vor. Von Oktober bis März in der Regenzeit also zieht es die meisten Zebras in den westlichen Nationalpark. Große Teile der Etosha-Senke sind dann überflutet. Mit Antilopen, Giraffen und anderen Savannenbewohnern durchstreifen sie dieses etwas üppiger bewachsene Randgebiet. Erst wenn dann die Senke langsam wieder austrocknet, wandern auch die Zebras wieder ostwärts. Brunnen und andere künstliche Wasserstellen bereiten den Tieren im Nationalpark vor allem während der Trockenzeiten bessere Überlebenschancen. Doch dieses Wasser brachte ihnen nicht nur Gutes. Viel zu viele Zebras kamen gut über die schlechte Zeit. Das karge Weideland von Etosha kann sie gar nicht alle ernähren. Etwa 10.000 Zebras gibt es hier. Und die Nationalparkverwaltung muss nun die Aussiedlung von etwa 1.500 Tieren erwägen. Bei ihren Wanderungen zu besseren Weidegründen bleiben die gestreiften Vierbeiner natürlich nicht nur unter sich. Gnus und Springböcke leisten ihnen Gesellschaft. Nahrungskonkurrenten sind sie nicht. Jede Tierart frisst andere Gräser. Jetzt haben die Zebras auch eine eigene Eskorte. Gabelschwanzdrongos heißen diese kleinen, respektlosen Gesellen. Eigentlich sind sie auf die Insekten aus, die von den Hufen der Zebras aufgescheucht werden. Am praktischsten ist da natürlich ein Plätzchen mit Übersicht. Das Schwanzschlagen nehmen sie dafür in Kauf. Die Drongos sind nicht die einzigen Plagegeister der Zebras. Zecken und allerlei anderes Kleingetier jucken und zwicken. Dagegen helfen nur ordentliche Staubbäder. Aber nie sieht man alle am Boden. Immer halten einige Wache. Zebras verwenden sehr viel Zeit für die Körperpflege. Durch das Putzen wird die Haut gesäubert und massiert. Unerreichbare Körperstellen pflegen andere Familienmitglieder. Das festigt die Beziehungen untereinander. Ernsthafte Kämpfe gibt es zwischen den Hengsten nur um eine junge Stute. Wenn sie heiß sind, haben sie bald mehrere Bewerber. Solche Zwistigkeiten gleichen aber oft eher einem Ritual. Gefährlich wird es erst, wenn die Gegner scheuen und mit den Vorderhufen zuschlagen. Bis zum Tode aber kämpfen sie nicht. Die Stuten bringen rasch ihre Fohlen in Sicherheit, auch sie sind wehrhaft. Will ihnen ein fremder Hengst zu nahe kommen, wissen sie sich zu helfen. Die jungen Hengste lassen sich auf den Händel miteinander ein. Bei ihren Kampfspielen geht es darum, den Partner zu zwicken, ohne sich selbst zu entblößen. Deshalb dieses abwechselnde Schützen und Wegnehmen der Vorderbeine und das Einknicken im Handgelenk, wenn der Partner angreift. Diese Spielerei hat auch eine ernste Seite. Es bereitet sie auf die Stellung als Familienoberhaupt vor. Sie üben sich für die Zeit, da sie selbst für ihre Stuten kämpfen müssen.
Der Kampf endet wie gewöhnlich, der Verlierer trottet davon. Bei der nächsten Gelegenheit werden sie wieder ihre Kräfte messen. Nur die kampferfahrensten und temperamentvollsten können später eine Herde zusammenhalten und vor fremden Hengsten schützen. Dunkle Wolken kündigen die Regenzeit an. Bald werfen auch die meisten Zebrastuten ihre Fohlen, denn die Zeit der fettesten Weiden bricht an. Den Regen lassen sie still über sich ergehen, die Ohren vorsorglich etwas nach unten gezogen, damit das Wasser nicht hineinläuft. Fast über Nacht sprießt jetzt frisches Gras. Wenn die Stuten ausgiebig weiden, haben sie auch viel Milch für ihre Fohlen. Es ist März und die großen weidenden Herden zieht es wieder zur Etosha-Senke. Überall gibt es Gras in Hülle und Fülle und gelbe Blumen, wo sonst alles trist und eintönig erschien. Die Savanne ist kaum wiederzuerkennen. Jeder hier genießt auf seine Art die frische Pracht. Wo verschiedene Tierarten in einem Lebensgebiet vorkommen, scheinen sie auf vielfältige Weise Nutzen aus diesem Nebeneinander zu ziehen. So sollen sich das Hörvermögen der Zebras und das Sehvermögen der Giraffe auf ideale Weise ergänzen, so dass es unmöglich sein soll, sich ihnen unbemerkt zu nähern. Zebrafohlen kommen früh am Morgen zur Welt. Dieses wird etwa eine halbe Stunde alt sein. Es ist noch ziemlich wackelig auf den starksigen, unbeholfenen Beinen, aber es hält sich tapfer. Eine Stunde später ist es völlig trocken und kann der Mutter folgen. Das ist lebenswichtig für Zebrakinder. Denn sie müssen laufen können, um ihren Feinden zu entgehen, und sie haben viele Feinde. In den ersten 3 bis 4 Tagen vertreibt die Mutter jedes Lebewesen aus der Nähe ihres Fohlens, auch die Stuten der eigenen Familie. So lange braucht das Kind, die Mutter genau kennenzulernen. Es würde sonst auf jedem anderen Lebewesen folgen. Später gestattet die Stute dann Verwandtenbesuche. Fast 12 Monate lang war die Stute trächtig. Während der nächsten Woche wird sie sich wieder paaren. Bis an ihr Lebensende kann sie Jahr für Jahr ein Junges gebären. 7 Monate lang säugt sie ihr Kind, dann wird es entwöhnt. Die erste feste Nahrung nimmt es übrigens schon mit einer Woche auf. Spielen gehört zum Zebraleben wie Grasen und Trinken. Wenn es in der Familiengruppe zu wenig Spielgefährten gibt, dann ist das für Junghengste ein gewichtiger Grund, sich anderswo Freunde zu suchen. Sie bilden dann recht ausgelassene Junggesellentrupps, die in den Herden für allerlei Wirbel sorgen. Dieses Fohlen aber hat noch Zeit bis dahin. Sobald sich die neue Regenzeit ankündigt, wird es mit den anderen Zebras hier in den westlichen Teil des Nationalparks ziehen. Es wird seine Zebra-Verwandten, auch die Bergzebras, kennenlernen und erfahren, dass im Leben eines Zebras kein Tag dem anderen gleicht. Zwar wird ihm eine immer wiederkehrende Palette ähnlicher Anforderungen abverlangt. Rivalen sind zu schlagen und tödlichen Gefahren ist zu begegnen. Doch wenn es Glück hat und wendig genug ist, kann es dabei 20 Jahre und älter werden, selbst viele Kinder zeugen und auf diese Weise für den Fortbestand der Steppenzebras von Etosha sorgen.